And now, to stay on track, we will be moving into the next presentation. So I'd like to introduce Dr. Matthew Busse to present the CEDAR curation and database structure. Thank you so much, Matt.
Great. Hello. I'll share my screen now. Alrighty, let's get started. Okay, my name is Matthew Busse. I'm a senior curator working on the CEDAR database, and today we'll be going through a brief overview of the curation process. Curation is the process of transforming unstructured data from the scientific literature into structured, searchable, curated data available in the CEDAR database. Papers of interest are identified from the scientific literature, then a team of curators works to extract, format, and enter the data into the database, resulting in a single resource of collated, searchable data that allows users to search for data relevant to their own research. Let's look more closely at this first step of identifying relevant papers. This is done using our custom-made biweekly automated query that scans through PubMed looking for potential papers of interest. Then those potential papers are sent to an automated text classifier, which sorts through at a more granular level, searching for uncuratable papers that are lacking critical information. Remaining potential papers are then classified into categories. The next step, a senior curator reviews a category of papers, providing human-level confirmation that the paper is actually curatable. And then individual papers are assigned to curators for manual curation. And throughout this talk, we'll be looking at an example paper to help bring the process to life. You can see the PubMed ID listed here. And this is a cancer paper that I recently curated, which is now available in the CEDAR database. Manual curation begins with the curator reading through the paper, searching for epitopes that meet the minimal inclusion criteria for the database. And these criteria are, it must be a linear peptide smaller than 50 amino acids in length, or a discontinuous epitope containing individual residues which have been shown to be crucial for immune recognition. Furthermore, in order to be curatable, an epitope must be tested in an assay as either an immunogen or an antigen. And we'll go over specific definitions of these in a little bit. And additionally, there's additional minimal information required, such as the sequence of the epitope, and for the assays involved, information such as the host, outcome, etc. So in our example paper, you can see in the methods section, there was a epitope sequence listed, and there was at least one curatable assay present in the paper. So that signaled to me this paper is valid for curation. And so these unstructured data from the paper were then extracted and entered into the data as structured data. So this process of mapping data from the unstructured data from the literature to the data map is a big part of what we do. And as you can see here, this is a highly simplified schematic of our database, and it's organized around epitopes and assays. So a curatable paper will have one or more epitopes within it, and each of those epitopes will be tested in one or more assays. There are a few critical fields required for every assay, and we'll go through those now. First is the host. This is the species producing the immune response. The immunization, which is the event which induces an immune response. Another key field is the antibody or effector cells, which represent the mechanism of the immune response. And finally, the antigen of an assay is the entity that is recognized by the immune response. So in our usage, an immunogen is what induces an immune response, whereas an antigen is what is recognized by the immune response. And for completeness, our definition of an epitope is the specific molecular structure interacting with the immune system. So let's look at an example of data entry. And this is done using our custom-built in-house data entry portal. Here you can see a screenshot of our modify epitope screen. Once again, here is the epitope of interest from the methods of our example paper. And here you can see the epitope sequence has been entered into the database, as long as additional information about the peptide. And each peptidic epitope is assigned to a protein ID of an existing protein from GenBank or SwissProt. So this links all peptidic epitopes to existing proteins that have already been identified. Now let's look at data entry for assays. So once again, here is an example assay from our example paper. In this case, it's a dextramer assay. And here's a screenshot of our modify T-cell assay screen. Here you can see the critical information has been entered. The host is entered as human. Here you can see the immunization has been captured as occurrence of metastatic melanoma. And this field shows the entry of the assay itself. Dextramer assays are curated as multimer/tetramer assays. Here you can see the outcome of the assay, which is positive in this case. And these fields capture the effector cells of the assay, in this case, CD3 positive T cells from blood. And finally, the antigen for this assay is the epitope itself. Whenever possible, data are entered using controlled value fields, which means that the appropriate descriptor of the data is selected from a drop-down list of available standardized terms. And this goes a long way towards ensuring that data are curated consistently from paper to paper and curator to curator. However, we also have a limited number of free text fields, which are necessary to allow curators to capture any additional data that doesn't easily fit into the controlled value fields. And it's this combination of controlled value fields with free text fields that enables to ensure consistent curation of papers, but also allowing the flexibility necessary to accommodate the scientific process. One tool that helps us do our job is the Curation Manual. And this is an online wiki of rules governing how data are mapped to the data structure. We have a general curation manual, as well as individual manuals specific to individual diseases, which provide additional details about how to curate situations specific to those diseases. And this is an online document that's updated as needed as we encounter new curation situations that require new rules. Another tool that helps us do our job are a series of ongoing field-level data validation checks. So several fields have automated validation checks that run as soon as data are entered into the field. For example, when a peptide sequence is assigned to a protein ID, if that peptide sequence is not actually found within the protein, it raises this validation error, warning the curator that a different protein ID is needed for this peptide. Once the initial curation is completed, then the curation and the paper go to a senior curator for a round of internal peer review. So the reviewer looks at the paper, looks at the curation, and provides human-level confirmation that the data have been accurately mapped to the data structure. And this can be an iterative process if needed, with rounds of back and forth between the reviewer and curator. And once the reviewer signs off on the curation, there's one final level of curation level validation checks, which looks at higher level validations, such as the relationships between epitopes and assays. For example, if a epitope is entered but no assays are assigned to it, it raises a warning flag, warning the curator that this epitope needs assays assigned to it. So once all of these curation level validation checks are finished, then the data is published to the public-facing CEDAR database. So a search of the database for our example PubMed ID will return one epitope used in 3 assays. And here you can see our multimer/tetramer assay that we looked at from our example paper. Data are published to the database on a weekly basis. And since 2021, the team has curated roughly 2,500 cancer papers since the start of the project in 2021. And current year papers, currently 2025, are curated as they are published and identified by our automated query. And we are also going backwards through time to curate the historical cancer data. We've currently made our way back to 2012. So that is a brief overview of the curation process, and we'd be happy to take your questions now.